So yes, I'll get straight to MONOCLE. MONOCLE stands for multi-scale observation networks for optical monitoring of coastal waters, lakes, and estuaries. It is coordinated by the Plymouth Marine Laboratory, which, although the name suggests that our focus is marine, we do quite a bit of work across the catchment, lakes and rivers as well, because basically that is where the scientific challenges are still very strong when it comes to optical remote sensing. We've got 11 partner institutes, and including some small companies, some SMEs involved in the project. All but one are in Europe. We work with Tafiri, which is in Tanzania, and they're one of the test sites for some of our citizen science objectives. And I will highlight some of the developments by each of these organizations, by some of these organizations in this talk. Now, I've divided this into two parts, because if I go straight into our sensor development and our challenges, then some context will be missing. So I'm actually starting from what we want to ultimately achieve, and that is a consistent and high quality, or perhaps I should say known quality, of the remote sensing through satellites of water quality in water bodies, in terrestrial water bodies. So this first part is collecting some existing approaches, and I will start with this survey that we did at the start of the MONOCLE project, actually. So we had always intended to be a little bit led by user requirements and not just our own development interests. And we sent out a survey to what we call water quality practitioners. So anybody working on water quality, whether it be research, technicians, developers, or water management authorities. We got quite a nice response, something like between 120 and 150 responses in a fairly short time. So it was really nice to see. And they were also from all over the world. And we asked many questions, and at the bottom right, you can see a link to our project website where actually all the survey responses are available, and you may reuse them as well. So they're in a PowerPoint format there. But I'll highlight just 2 or 3 results that we had. So first we asked, when it comes to sampling water and sampling the variable that you find particularly interesting, so we did not specify this, then first of all, how frequently do you currently sample this? What is the minimum required sampling frequency to meet regulations? So for example, the Water Framework Directive. And finally, what you consider would be an adequate sampling frequency to capture natural variability in the system. And you can see that the responses peak at different intervals. So the minimum required by law is fortunately below what we actually achieve, and that's usually quarterly to monthly is considered sufficient. The current practice ranges a little bit wider, but in some cases it is done weekly, daily, or even more frequently. But the desired frequency was almost in all cases a step higher. So most people responding said, well, really, to understand the system best, we should be capturing this information at least weekly, but preferably daily or hourly. And that is not yet achieved. We also asked, well, what are the variables that you consider most important to be monitored in your water bodies? And this wasn't a surprising result. We know that nutrients are of prime importance to predict the health of aquatic ecosystems. Eutrophication is still one of the major challenges in maintaining water quality. Dissolved oxygen also describes the biological component of the aquatic systems. And then there were some proxy variables such as chlorophyll, or pH, turbidity, and individual nutrients mentioned salinity in some cases. And we just went through this and said, okay, well, some of these you cannot complement using optical observations, let alone satellite remote sensing, but some are. So the ones in green here, they are achievable to an extent with complementary satellite observations. So then if we combine these results only, then we can look at what satellite observation is currently able to offer as by means of a solution to fill in data gaps, for example. And in terms of frequency, we can now safely say that what we call optically complex water bodies, so those that are inland, influenced by terrestrial runoff, including rivers and lagoons along the coast as well, They can be monitored up to daily with what currently is available in satellite assets. In terms of continuity, which is another important factor for monitoring, we can safely say that through the European Copernicus framework, the same sensors that are currently being used will be able to provide data over the next 20 years, because the follow-on sensors are already ordered and will be launched when needed. And in terms of timeliness, in terms of being able to respond to issues with water quality, we can generally say that useful satellite products can be made available within sort of the span of a day. If it's an emergency situation, then things change a bit and it becomes even quicker. And just to show you that this background picture here is an actual result from the Copernicus Inland Water Quality Service, which is available for now over 4,000 lakes worldwide. And this product is the Trophic State Index. And you can see that quite a lot of the Canadian lakes, there's some cloudiness in here as well, obscuring some of the results. Quite a few of even quite small Canadian lakes here are included in the service already, and it's global. It's nice to look at Canada because there are so many. So I won't explain every detail of remote sensing, but just to get us to what the main challenges are, what we are actually observing with satellites, or in situ systems for that matter, is the color of the water. But that is the color of the water in a sort of normalized principle. So we look at the reflectance, that is the radiance coming out of the water, as a function of the sunlight that is reaching the water. And then there are some interferences. Some of the light that we can see at the satellite sensor has actually been reflected in the atmosphere. And some of it has been absorbed in the atmosphere. There is some interaction at the water surface, and then a smaller fraction of the sunlight actually makes it into the water column and back out again. And then as it comes back out, it has to pass again through the atmosphere. So obviously the atmosphere can have quite an influence on the signal observed from space. What we're actually interested in is only the light that interacts internally in the water column. And there it can be absorbed and scattered by phytoplankton. It can be absorbed by dissolved substances, which have a clear link to the carbon cycle. It can be absorbed or scattered by other suspended particulates from bacteria to zooplankton to fish, if you like. And finally, some of the light reaches the bottom and will be absorbed there. So this is a fairly complex picture. If this was the ocean, it would be a lot simpler. In the ocean, we know it's mostly clear water, and phytoplankton are the main source of primary production. So also the other components are related to the biomass of phytoplankton. When it comes to inland waters, we can't say that. Therefore, we call these systems optically complex. And when we measure the water leaving radiance from space, and we try to correct for the effect of the atmosphere, then ultimately we still have what we call the inverse problem, which is to attribute the color of the spectrum of light, the spectrum of light that we have observed, to these absorption processes and scattering processes in the water column. Okay. Of course, people have been working on this problem for quite some time, and in fact for longer than the first satellites appeared in space. And most of this work has actually originated in inland water bodies. What we currently have is quite a nice series of satellite systems, which all have to be treated in their own way. They have slightly different observation wave bands. They observe different parts of the blue, the green, the red, the infrared part of the light spectrum. And we've got this, well, I wouldn't say it's complex. It's fairly linear, actually, this processing scheme where we take in satellite passes, we do various corrections, we do an atmospheric correction. And then one of the first steps we do after the atmospheric correction is complete is we run a classifier that separates each observation, each pixel, each observation day into a similarity to set optical water types. And that is used to inform the choice of algorithms that we use to then infer what the chlorophyll concentration might be, or what the effect of suspended matter might be. Now it's interesting to note that currently just the water reflectance, so that is the actual color of the light coming out of the water, is an essential climate variable. So that's a purely physical variable that we're trying to report in terms of long-term climate data sets. So that also means that there is some funding going into collecting this through, for example, the European Space Agency Climate Change Initiative. When we speak to most of our users, such as water quality management authorities, biologists, limnologists, the main interest is actually in quantities derived from the reflectance, and that is things like chlorophyll or suspended matter. So a large group of people who rely or who would like to use remote sensing data sets don't really care so much about the reflectance, but care a lot about the quality of the derived products. And they are related, but not fully dependent processes. So I'm going to jump first to the current state of the art in producing the chlorophyll variable. That is the main quantity that is needed to understand the ecological health of the biological health of aquatic systems. It is an indicator in the Water Framework Directive, so it is fairly central, and it has been measured in situ for 3 decades at least. So what we see is various algorithms exist to translate the reflectance spectrum. Here are 4 results on the left into a chlorophyll estimate, but they don't work equally. So for example, this OC2 algorithm, OC stands for ocean color, and it was really designed to deal with fairly clear ocean waters. Then there are 3 algorithms here that all work in another part of the spectrum and have slightly different assumptions on what to expect there. None of the algorithms works particularly well over the whole range. So what we do is, and that's indicated by the different colors here, is we assign the algorithms that work best to optical water types. And then based on the similarities of an observation with that water type, we do a weighted average to come up with a blended result. And that result looks a lot better than the individual algorithms. So that's how we currently deal with going from the reflectances after atmospheric correction to producing a chlorophyll estimate. And in this case, we can build this on what we call a match-up data set, so co-occurrence between satellite and in situ observations of more than 2,500 occurrences. And that gives us a reasonable tool to observe this water quality variable at the global scale. So we don't necessarily rely on having in situ measurements from a lake in China to know that based on the appearance of that water body, we have a suitable algorithm, because we link it all to the optical water types that have been registered elsewhere and for which we have this type of validation. We would love to have more of this though, because there are still large uncertainties, and we would like to relate that uncertainty to, for example, what sort of sediment type there will be, or what sort of surrounding land use there might be, or how actually the in situ data have been observed, because there are some methodological differences. And I wouldn't say that all of the scatter here is necessarily caused by remote sensing uncertainties. Some of it will be in situ as well. Okay. When we map this procedure using here in the columns 4 different algorithms types, and we then through a weighted averaging create a single map, you get a reasonable response, so a natural looking response for quite diverse water bodies. So there's 2 large lakes in Sweden here, so a lake in the Rift Valley in Kenya, and a very eutrophic lake in Taihu. So these don't show very strange occurrences anymore, whereas if we applied one single algorithm, we would see some weird behaviors. We can go as far as estimating the product uncertainty based on the validation that we have done previously. So we can also map that for each pixel. Unfortunately, that product uncertainty results in something in the order of 50%. And that is too high when we think about the requirements for climate data records. So the state of the art, as we have implemented it at the global scale, doesn't actually quite meet the target requirements for global observations. And that is where we are now. And that is, I predict, where we will be for some time. So our target is much stricter than what we can currently deliver. And the reason this won't change very quickly is because we can't collect a lot of in situ data over a short time to inform the algorithm developers how to do a better job. And at the same time, the satellites that we have in space are not changing very quickly. So we cannot easily design a more suitable satellite and launch it next year. This process takes 5 to 10 years sometimes. Okay. Between capturing the signal at the top of the atmosphere by the satellite sensor and going all the way through a series of algorithms and model assumptions to derive something like chlorophyll concentration, we have the reflectance itself. And we know that this is the most uncertain step of the process. So here you can see for actually the best atmospheric correction processor we have, how the remote sensing reflectance that we observe from the satellite corresponds to the in situ observed reflectance spectrum. And the first thing to observe is we don't have a lot of data points on this, less than 300, which is really not a lot to try and describe how this concept performs at a global scale. So here we are taking, you could say, a big risk. Now, fortunately, we have already seen that despite what might be an over or an underestimate, what might be systematic biases, we are able to correct for a lot of this uncertainty when we go all the way to the chlorophyll algorithms, because they are individually optimized for all these optical water types. So we can circumvent some of this, but we should not forget that the reflectance itself is the physical parameter and is the essential climate variable. So this is what we actually would like to get right. And then we can tune the downstream derived products again to still make sense. So here's the largest challenge. So my conclusion here is that despite this somewhat disappointing result, it's based on very, very little in situ data. So that means two things. We don't actually know if it is a consistent underestimate, because maybe if we sample the next 5 legs, it will be a lot higher, or in a different season. And also we cannot inform the people developing atmospheric correction routines how to do their job better, because these are really only, 10 lakes or so that we are basing this information on. So this is the priority. Getting more in situ radiometry done is the main concern. So we are trying very hard to convince government agencies, for example, that it is worth doing this. Although this does not give a chlorophyll A observation, improving this step of the process means that we can be less uncertain about the products that we deliver for water quality reporting, for example. Okay, and then there's one other aspect to the radiometry challenge, and that is that there are multiple sensors and none of them last forever. So we're showing here 3 different sensors over Lake Michigan at 5 different reflectance wavebands. And you can see that in the light blue, there's actually the longest going sensor currently. That is the MODIS sensor on the Aqua satellite by NASA. It gives a quite steady seasonal response. It has daily observations. It's a very steady sensor, but it has a limited set of wavebands. It is not ideally suited for lake observation. So very often we only refer to the satellites marked in black and red, and that is the MERIS sensor on the Envisat satellite, and the Olchi sensor, of which we currently have 2 on the Sentinel-3 satellites. And they seem a bit more noisy, but that doesn't necessarily mean it's worse. It could also be that they are better at picking up variability than the MODIS sensor is here. So what you see here is maybe some trends, which could be a degradation of sensors, or it could be a change in water quality, or it could be differences in how we manage to do the atmospheric correction based on what sensor we have. And in order to come to the changes in water quality part, which we really need, we need to be able to compare these signals to signals on the ground. Okay. So in summary, for this first part, satellite observations have come a long way for inland water quality monitoring, but we have major challenges stemming from how to separate the atmospheric and the water signals. I haven't talked about this, but accurately detecting the difference between clouds and ice and snow always depends on what the capabilities of the sensor are. When we go near land, the uncertainties of the signal from the water and the signal from the land mixing in the atmosphere becomes quite a difficult issue, which means that we are actually better at remote sensing large lakes than very small lakes, because the land is closer in small water bodies. And finally, we have to deal with the optical diversity of water bodies. We cannot apply one algorithm to predict biogeochemical properties for all water bodies. And in fact, the optical diversity can also be within a water body or change between seasons. So we have to always have a dynamic approach to this. And this part we have covered quite well now, I think, with our intermediate step of mapping water types and matching algorithms. Okay, so what we really need from in situ activities is to add radiometric reference measurements, to combine that as far as possible with traditional biogeochemical sampling, so pigments, dry weight, etc., and maybe taxonomy, but also to have an optical component of this, because that's what we get from the satellites. That will be a better match. And we need to combine these observations both in open water, where we can do a fairly confident comparison to satellite observation, but also near the shore, where we know that other factors come into play. And none of these things are typically included with any of the ongoing statutory monitoring. So while there is a lot of data in situ coming out of many countries, especially Western countries, we cannot necessarily use it all. Okay,